Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, welcome to part 2 of chapter 5 of our Bauman textbook covering photosynthesis. Let's go ahead and get started. Hopefully you've had a chance to watch my review of photosynthesis. That's going to be in a card at the top right of your screen right now. Please make sure you've watched that video on photosynthesis because that is a good basic overview. And this video, because this is a second year class, I'm going to assume that you understand photosynthesis already. And in this video, I'm going to be simply highlighting some of the more important things to know and also juxtaposing photosynthesis a little bit in prokaryotes versus eukaryotes. So let's go ahead and get started. If you remember, photosynthesis is the converse reaction. That means opposite reaction to cellular respiration. In cellular respiration, we began with, let me actually write it down for you because I want you guys to understand this. Here, go ahead and modify your own notes if you would like. Let me show you what I'm talking about. For cellular respiration, remember the equation was C6H12O6. That's glucose, one glucose molecule, and 6O2, 6 oxygen molecules. And that became, those are the reactants, which then became 6 CO2. That's why you breathe out oxygen, you breathe in oxygen, you breathe out CO2, right? That's why you breathe out CO2, a much higher concentration of CO2. And there was also another waste product, 6 water molecules, H2O. And the useful product was ATP. Remember, there were 32 ATP in prokaryotes, 30 net ATP in eukaryotes. But we were talking about ATP energy. Okay, so that's cellular respiration, correct? Now, what did I say photosynthesis is? Photosynthesis is the converse reaction. That means the opposite reaction. So if we were to talk about photosynthesis, let's go ahead and do that. Let's talk about photosynthesis. It is actually the converse reaction. What does converse mean again? Opposite. So the arrow is going the other way. You see that? And instead of ATP energy as a reactant, you've got a different type of energy. You've got light energy or sun or light energy. Does that make sense? So you're taking sunlight energy, you're taking water, you're taking CO2. And those are the reactants you need for photosynthesis, and you're producing oxygen. This is why people say that plants produce oxygen for us to breathe. And you're producing sugars and other carbon-based molecules, other organic molecules. But mainly, we're going to be discussing sugar. Okay, so how does it do this? Again, like I said before, hopefully you've watched the video that I told you to watch. Covering photosynthesis, but remember that it all depends on these chlorophyll molecules, these pigment molecules, including chlorophyll A, chlorophyll B, xanthophyll, carotenoids, etc. There's many different types of pigment molecules. And these pigments, they get excited by light, and then they pass on that energy from sunlight. Okay, so let's talk about the difference between prokaryotes and eukaryotes. Remember, we want to know what is the main difference. Well, in eukaryotes, this is something you've studied. You've studied photosynthesis in eukaryotes before. In eukaryotes, remember, Where does photosynthesis happen? In a special organelle called the chloroplasts, right? There's a chloroplast. Chloroplasts are membrane-bound organelles. So because of that, prokaryotes do not possess chloroplasts. Again, in eukaryotes, photosynthesis happens in the chloroplasts, and there are these inner membranes, this folded inner membrane called grana, and that is where photosynthesis happens, in the grana, the thylakoid membrane, right? But in prokaryotes, again, they do not possess chloroplasts, so the place where photosynthesis happens is actually an invagination of the cytoplasmic membrane. That means that the membrane, the cytoplasmic membrane, or the plasma membrane of the cell, actually folds inwards to form grana-like substance structures inside of the cell. Okay, so this is what we're talking about. And again, you can see what the pigment molecules do. Remember, pigment molecules become excited by photons of light. What does that mean? That means their electrons go to a higher excited state, a higher energy state. And when those electrons come back to the ground energy state, that's when energy is released from the pigment molecule. Okay, you should already know about photosystem 1, photosystem 2, and how they work with the light dependent and light independent reactions. Remember the concept of cyclic or non-cyclic photosynthesis, or light reactions of photosynthesis. Remember from my previous video how light hits a pigment in the photosystem, and that pigment becomes excited, which excites its neighbor, which excites its neighbor, and the excitement eventually reaches the central chlorophyll pair, or the special pair of chlorophyll. In photosystem 2, remember, it was called P680, and in photosystem 1, this is called P700. Here it is again, this purple structure with everything inside of it, all the green structures inside, the blue structure inside this, it looks like an apple. This represents the photosystem. When light hits a pigment in the light harvesting complex of photosystem, that pigment right there, let's say that pigment is chlorophyll A, it could be chlorophyll B, it could be xanthophyll, it could be carotenoids. That pigment becomes excited, which can then excite its neighbor, excites its neighbor, excites its neighbor, excites its neighbor, excites its neighbor. The excitement reaches the special pair of chlorophyll A. That special pair releases electrons. How can it do that? Well, because it switches to a highly negative E sub O. Remember, E sub O stands for standard reduction potential. The standard reduction potential switches negative, electrons leave the central chlorophyll pair, the electrons go to this box. This box is called the primary electron acceptor, at which point the electrons can flee, can leave the photosystem altogether. Okay, and this was a nice overview of the light reactions of photosynthesis. Remember, the story begins at photosystem 2. Light strikes a pigment, exciting the pigment, which excites its neighbor, which excites its neighbor, which excites its neighbor, which excites its neighbor, which excites ultimately the central chlorophyll pair. And remember, the central chlorophyll pair on photosystem 2 is called P680. P680 switches to a negative E sub O, or standard reduction potential, releasing the electrons to the primary electron acceptor. Those electrons follow the yellow arrows. The electrons go through PQ, plastoquinone, through cytochrome complex, PC, plastocyanid, and to photosystem 1, okay, to the central chlorophyll pair on photosystem 1, which is called P700, okay? Now, the electrons moving through this structure, PQ, cytochrome complex, and PC, this is called the electron transport chain. This is an electron transport chain. The spontaneous flow of electrons through this electron transport chain provides the energy that cytochrome complex needs in order to pump protons across the membrane. And remember, this membrane is the thylakoid membrane in eukaryotes. However, this membrane is the cytoplasmic membrane, the plasma membrane, the cell membrane in prokaryotes. Okay. Now, The electrons sit here on P700 of photosystem 1, and they cannot leave until light strikes a pigment on photosystem 1's light harvesting complex, exciting that pigment, which excites its neighbor, excites its neighbor, excites its neighbor, excites its neighbor. The excitement reaches P700. P700 switches to a negative E sub O, standard reduction potential, and then the electrons go on their merry way through FD and to this enzyme called NADP plus reductase. It is the enzyme that reduces or gives the electrons that just came to it, gives those electrons to NADP plus along with a random proton, which forms NADPH. So you reduce NADP plus to NADPH with the incoming electrons from this light harvesting complex process. The enzyme responsible is called NADP plus reductase, and now you have the NADPH that you need. Now, while the electrons flow through the electron transport chain, remember what was happening. Protons were being pumped into the thylakoid space in eukaryotes. However, in prokaryotes, protons are being pumped from the outside of the cell into the cytoplasm of the cell. Does that make sense? Now you get a high concentration of protons inside of the cells, or a high concentration of protons inside of the thylakoid space, right? The thylakoid space of eukaryotes, okay? At which point the protons force their way out through chemiosmosis, through ATP synthase, causing ATP synthase to spin and causing phosphorylation of ADP. ADP gets phosphorylated to ATP. You see ADP along with an inorganic phosphate join forces to become ATP. So now you have ATP, you have NADPH, those head off to the Calvin cycle, ready for powering the Calvin cycle. They're used by the Calvin cycle in order to form these sugars. Now, how do we restart this process? Well, the electrons that left P680 at the beginning of my story, they need to be replenished. And where do they come from? They come from water molecules, water. This is why you have to water your plants. This is why water is a water is a reactant of photosynthesis. Why is water a reactant of photosynthesis? Well, because it's needed to donate its hydrogens. You see that? Donate its electrons to reload P680, which is at this point called P680 plus, which is, by the way, a highly oxidative force. P680 plus, because it gave away those electrons, is now P680 plus. It went from P680 to P680 plus. P680 plus is a highly oxidative form of this central chlorophyll pair. It has a very, very positive E sub O, a high standard reduction potential, so it can steal electrons from water. Those electrons are then sped through this process upon more light striking this complex photosystem too. All right, so water loses its electrons to the central chlorophyll pair P680 plus. P680 plus goes back to P680 and the process starts over again. Now, what's left over of water when you strip it of oxygen? When you strip it of its hydrogens, water gets oxidized to oxygen. Water loses its electrons, right? So water gets oxidized to oxygen. This is why oxygen is a product of photosynthesis. So again, let's do an overview. What are the products of linear electron flow during the light reactions of photosynthesis, which is what you see here? The products are NADPH, ATP, and oxygen. And what was required for the light reactions? Water and light, correct? And again, what I just explained was linear electron flow during the light reactions of photosynthesis. What does that mean? Linear means the electrons started on water and they make their way in a linear fashion to NADP plus to form NADPH. So those electrons on NADPH started on water, and they made their way there in a linear fashion through this process. So what's cyclic electron flow? Cyclic electron flow means that once the electrons reach P700 on photosystem 1, then every time light strikes photosystem 1 and causes P700 to release electrons, the electrons, instead of going to NADP plus reductase, the electrons double back through cytochrome complex and back to P700. Again, upon more light, the electrons double back through cytochrome complex and back to P700. So this is cyclic electron flow, which means what I want you to understand is during cyclic electron flow, that means that the electrons are only going through photosystem 1, back through the electron transport chain, photosystem 1, back through the electron transport chain, and this happens over and over again as long as light is present, right? So if this is the case, what is the only product of cyclic electron flow? Think about it. What should be the only product if the electrons are doing this over and over again? That's right. Think about it. That's right. So if the electrons are constantly flowing through cytochrome complex, that means they're providing the energy for cytochrome complex to actively transport protons into the thylakoid space, and those protons can then force their way out into the stroma, forming ATP. And do you all remember how this ATP is formed? What is this ATP formed by? What process? Remember, is it substrate level phosphorylation? Nope. Is it oxidative phosphorylation? No. It's called photophosphorylation. This ATP is formed by photophosphorylation. All right, great. So this is just a review of what we discussed: linear electron flow, cyclic electron flow. So now let's talk about the light-independent reactions, aka the Calvin cycle. The Calvin cycle happens in 3 steps: fixation of CO2, reduction of that CO2 to sugar, and regeneration of the RuBP, Ribulose Bisphosphate. Again, this was covered in the last video, but I'll briefly touch on it again for this video. If this is at all confusing, please go back to that other video that I linked at the beginning of this video. Again, the whole point of the Calvin-Benson cycle is to make sugar. How do you make sugar? You need the recipe. What's the recipe for sugar? Sugar consists of carbons, hydrogens, and oxygens, right? So let's make sugar. We're going to get our carbons and oxygens from CO2, from the environment, from the atmosphere. CO2 enters the Calvin-Benson cycle. CO2 is joined to this molecule. This is called the CO2 acceptor, ribulose bisphosphate, or RuBP. Then it is cleaved in half to form phosphoglyceric acid, or phosphoglycerate. This phosphoglycerate is then reduced by NADPH. Remember, NADPH, these are the H's. These are the electrons from our water from the previous step. We're going to give those H's, we're going to give those electrons to our organic molecule here. So now we're going to have not just C's and O's, carbons and oxygens, we're going to hand it the rest of the recipe for sugar. H's, hydrogens. Hydrogens are going to be added onto our organic molecule here. And with the help of ATP, ATP is used to energize this process of giving hydrogens to the sugar, to the organic molecule, until we have what? We have C's, H's, and O's. We have glucose, C6H12O6. Did you see what happened? CO2 was taken out of the air. With the help of ATP, we gave that CO2 H's, we gave it electrons or hydrogens, and we end up with C's, H's, and O's, glucose. This is called reduction. And the first part, by the way, where we took CO2 out of thin air, we converted CO2 from a gas to a solid, right? This is called carbon fixation. So we have carbon fixation followed by reduction, which means giving that CO2 hydrogens or electrons more specifically, right? And then the last step is called regeneration of ribulose bisphosphate. We have to remanufacture this organic molecule, RuBP, ribulose bisphosphate, so that we can start the cycle over again. Just like at the end of the Krebs cycle, you had to remake oxaloacetate. At the end of the Calvin cycle, you have to remake RuBP, ribulose bisphosphate. And by the way, before I let you go, one more thing you should know is what enzyme is responsible for carbon fixation. What was the enzyme that attached CO2 to ribulose bisphosphate? That enzyme is called Rubisco. Rubisco. Okay, so Rubisco is the enzyme responsible for carbon fixation. Ribulose bisphosphate is, or RuBP, is the molecule that needs to be replenished at the end of the Calvin cycle. And again, the steps to the Calvin cycle are carbon fixation, reduction, and regeneration of RuBP, ribulose bisphosphate. What is required by the Calvin cycle? It looks like CO2 is required from the air. ATP is required from the light reactions, and NADPH is required from the light reactions as well. What are the products of the Calvin-Benson cycle? Glucose is the product of the Calvin-Benson cycle. And what enzyme is responsible for carbon fixation? That's right, Rubisco. Rubisco is the enzyme responsible for carbon fixation. Awesome. So I think that leads us to the end of the chapter here. Hopefully this was all a review for the most part of Biology 1406 with a little bit of prokaryotic versus eukaryotic mechanism sprinkled in. I hope this was an informative video. Let me know in the comments below if you have any questions, and I'll catch you guys next time.
Dr. D, 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 Dr. D.